Hello everyone, this is Dr. Maha Abdurrahman. I'll be doing some very important questions today about genetic engineering since I am having multiple requests from my students on these particular chapters. So I will be doing today a question explaining biotechnology and genetic engineering. And let's start with the definition to start with. What is genetic engineering? So we simply, what do I mean by genetic engineering? So genetic engineering is using genetics to produce a material or hormone or protein that is useful. How can I do that? Basically, I am taking or removing part of the gene. So I'm taking a gene, or I'm taking DNA, part of the DNA, because basically the gene is part of the DNA, okay, from one organism, right, or from one species, and then inserting this gene, okay, into another organism. Okay, why am I doing that and how am I doing that? This is the purpose of my tutorial today. So why am I doing that? Because this gene will eventually be used inside this new cell that has been inserted in to produce the protein or the hormone that I need. Okay, let me give an example for that to become more clear. For example, the production of insulin, the insulin that you see in pharmacy, okay, that the pharmaceutical companies sell. From where this insulin is coming, it is produced by genetic engineering. How this is possible? So basically, we need to remove the gene that is responsible for insulin production, okay, which as you see, I will explain it here on the diagram first. So this is the cell, that's the cell of the species or the organism that I want to remove a gene from. Obviously, this is the gene. I'm drawing with red. So this is the gene, and M is the chromosome, okay? L is the cell membrane of the organism that I need to remove the gene from. Basically, N is this particular gene, which should be here the insulin gene, right? And then I'm gonna insert this gene into the DNA of another organism, okay? Now, this other organism is bacteria. This is a bacteria cell. How do I know that's a bacteria cell? Because as you see here, this kind of DNA, this kind of circular DNA is called plasmid. This plasmid is circular DNA. It is only found in the bacteria. So basically, I'm removing this plasmid, and then I will cut through this plasmid. After I remove it from the bacteria, I will cut through it using enzymes. These enzymes act like scissors that cut through the DNA of that bacteria, and then I will insert my gene inside this plasmid, just like glue and stick, okay? So you're gonna glue this gene inside this plasmid, okay? How am I gonna do this sticking or this gluing? Using another enzyme. So basically, I'm using an enzyme like a scissor, and I'm using another enzyme like glue to attach this insulin gene into the plasmid. Okay, so now I have done this recombinant DNA. I will insert it again inside the bacteria. Then I will allow this bacteria to reproduce. When this bacteria why do I need first of all to allow this bacteria to reproduce? So I can have a lot of this recombinant plasmid. Then this bacteria will be placed in a tank, as you see here, allowing this bacteria some time to produce the insulin gene, because what's going on in this bacteria, the insulin hormone. So the insulin gene here inside this plasmid will be processed by the bacteria. This bacteria, when it reads this insulin gene, it's like a recipe. This gene, guys, is like a recipe, okay? So this bacteria will use this insulin recipe. Now it has this insulin recipe inside it, right? It has now this book of recipe. It will start to read and make this recipe. So production of insulin will be the product that I will get after a while. So I will get, so here in this tank, I will have insulin plus bacteria, right? Because I still have a lot of bacteria that produce this insulin. If I manage to remove this bacteria by filtration or any other way, I will have insulin ready to be used. So this is in a nutshell how genetic engineering is working. If you understand what I said now, the questions will be for you a piece of cake. So let's check here. So basically here they're asking you about using the letters from up to attain or to complete this table. So basically M here, M is the chromosome, the thread of DNA found in the nucleus. What is the section of the DNA removed from the human cell called? It's the gene. Gene is the section of the DNA removed from the human cell, and I'm gonna write a letter for this, which is, as you see here, M. The plasmid, as we mentioned, it's a circular DNA found inside bacteria, okay? Sometimes we refer to this plasmid as vector. What do I mean by vector? Vector means carrier, right? So you can write vector, you can write, and it is a circular, of course it's a carrier for the gene, right? For the insulin gene. So it's a circular DNA, okay? And we're gonna give that the letter, just let me move a little bit, Q. It was given the letter Q, okay? Then the type of the cell that is genetically engineered is the bacterial cell, okay? So if it was a bacteria, sometimes they're using yeast for that. You can either use bacteria or yeast, both are accepted, but I'm gonna write bacteria here because it is the one that has plasmid, as I mentioned. So it should be bacteria. And the letter here was letter R that was given, as you see here, letter R. And then a specific chain of amino acid coded by this DNA, by section of DNA removed from the human cell. What's the specific chain of amino acid? Is basically a protein, right? Coded by this section of DNA removed from the human cell. So what is this chain of amino acid, which is, as I mentioned, it is a protein? Well, this protein is the insulin. Is insulin a protein? Is insulin protein? Yes, insulin is a protein. Okay, insulin, but someone would say, but Miss, insulin is a hormone, as I know. Yes, and hormones are proteins. Da-da. So that's a surprise for those who don't know. Hormones are proteins. So you can write insulin, you can write protein. Both of them are accepted. And the letter was O. Okay, and so basically here, guys, it's just showing you, maybe I did not refer to this. This is the insulin protein. It's just showing you that it comes out after the cell process this gene, this insulin gene, it will make the insulin itself. Remember always the book of recipe. Where is the fermenter? It's P, right? Fermenter is P, which is a big container to have the bacteria produce insulin inside it. So I'm gonna write P. It's the container, a big container that bacteria can grow, or where bacteria can grow inside. Why do I need this bacteria to grow? To produce insulin. I hope this genetic engineering process was clear for you. And the coming question is asking that the genetically engineered cells, like this bacteria cell, they're obviously reproducing asexually, and they're bringing us back to the advantage and disadvantage of asexual reproduction for insulin producing for insulin production by genetic engineering. What's the advantage of using this technique, which is asexual reproduction of bacteria, to produce insulin? Now, that's a 3 marks question, so I need to go into some more details, okay? Basically, the first advantage when I talk about asexual reproduction compared to sexual, that it's rapid, it's fast. It's fast, it's rapid, okay? More rapid or faster than the sexual reproduction. So asexual reproduction is faster than sexual, okay, because there are no gametes involved. No gametes involved. Okay. The second advantage for this is that you're getting, in the asexual reproduction, you're getting exact copy. You're getting exact copy. You're getting a clone, right? You're getting a clone. Your exact copy, a clone of the bacteria you are using, or of the living thing you are using. Why this is important? Because basically I'm gonna get a lot of bacteria that have this recombinant DNA inside, which carries the gene of insulin, which can process more and more oxygen. Okay, so I'm gonna write that as well, that this will end up having many bacteria, so having many repeated cells having the same insulin gene. Okay, so this can lead to have a large amount or large quantity of insulin. Okay, I hope it was clear about the advantages of the asexual reproduction. And let's move to another question. Also, this question is kind of challenging for many students. That's why I'm gonna do it as well. Antibiotics are used to treat human diseases. Now, many bacteria have become resistant to the antibiotic. That's a hot topic that is very prevalent nowadays. Antibiotics, some antibiotics can no longer be used to treat certain diseases because bacteria are getting resistant. Samples of the bacteria were taken from person who had an infection disease. They were spread out onto 4 petri dishes of agar plates. 3 of these agar plates contain the antibiotic, 1, 2, and 3. So basically 1, 2, and 3, there is an antibiotic in these plates. Okay, and then they added some bacteria on these 3 plates. And then this plate, it has the bacteria, but there is no antibiotic, as you see here. Okay, and then the shaded area is showing you that there is a bacteria that is growing after a couple of days on the agar, of course, providing they're giving them the proper temperature to grow, and it's been left couple of hours or maybe overnight. And then the other plate, there is no bacteria. Now, no bacteria grew in the agar plate with antibiotic 2. What does that mean? No bacteria grew. What does that mean? That means simply that this antibiotic 2 kill all the bacteria. As simple as that. Yes, antibiotic 2 has killed all the bacteria, as simple as, and I get this mark. Now, bacteria grew in the agar plate with antibiotic 3. As you see, it's full of bacteria. What does that mean? That means that this bacteria that is fully grown is not affected by the antibiotic at all. This bacteria, all the bacteria here in this plate, all bacteria is resistant. Resistant is not affected to antibiotic. This antibiotic could not kill the bacteria. And I get my second mark. Let's move down to why only a small number of bacteria grew in antibiotic 1. There are some bacteria that managed to grow in the plate having antibiotic 1. That means some bacteria have some resistance to this antibiotic, not all of them. Well, these were the bacteria that were able to grow. Okay? Yeah, so that's it. How many marks do we have here? We have here 2 marks. So this answer would be enough. If you want to go further, if this question was a 3 or 4 marks, you would have to mention a reason. Okay? Why did this bacteria developed in a resistance to this antibiotic? Why? Why do you think this this bacteria developed resistance to antibiotic? What causes bacteria to develop resistance? It's and that's a very, very common name that we hear, mutation, right? When bacteria undergoes mutation, they can become more fierce, stronger, that they can resist the antibiotic, okay? So what I'm gonna say here in this question is that only small number of bacteria is resistant to antibiotic. Okay, as I mentioned, it's always lovely to mention the reason. So I would say because their genes has been mutated. Mutated means change. The genes has been changed, giving more favorable characteristic to this bacteria, and so that this resistant bacteria can grow and reproduce. Okay? Common sense, right? Now explain why it is important to carry out a test similar to that shown in figure 2.1 before giving an antibiotic to a person infected with bacterial disease. This is raising again the bacteria, the antibiotic resistant. I don't want the bacteria to become resistant to antibiotic, so I should make this kind of test to prevent the antibiotic resistance, to make sure I'm using the most effective the most effective antibiotic to kill this bacteria. Because if I'm not using the most effective antibiotic, not only this person will not be cured, also the bacteria can be more resistant or can develop resistance. So I don't want that to happen. Okay? So as I said, I'm gonna write the 2 points to find out the most effective antibiotic to kill all. I need to kill all the bacteria in that person. As I mentioned, not only to cure the person, so I would add to cure the person, and not only that, and also to prevent antibiotic resistance. I don't want antibiotic resistance. I need to make sure that I'm using the correct antibiotic, otherwise the bacteria may develop some resistance. Okay? Now, antibiotic resistance has become a major problem worldwide. As I told you, it is very, very important topic. in science nowadays suggest how the problem of antibiotic resistance can be limited, and that's a 4 marks question, and this question is really touching base with our everyday life. Whenever we go to the doctor, he is reluctant, or he should be reluctant to prescribe antibiotic until he is making sure that this is a bacterial disease, because prescribing antibiotic more often for viral infections or when it's not needed can lead to antibiotic resistance. Bacteria becomes mutated. Okay, that's why also I don't want that to happen. So other than making sure I'm using the antibiotic only for the bacterial disease, also I need to tell this people or this patient to make or to use this antibiotic for the full course. Otherwise, bacteria can get more fierce, can become resistant to the antibiotic. If I'm just giving the bacteria the taste of the antibiotic and then I cut this out again, bacteria can be develop mutation, can become more fierce, can develop antibiotic resistance. Let me make that, I'll add that into a bullet points, or the points that will have the mark is referring to how the problem can be limited. So prescribing antibiotic only when needed. So limiting that, antibiotic only, prescribing antibiotic only when needed. What do I mean by only when needed? That means not for viral infections, Also, we need to ask a patient to complete the course of antibiotic. We need to ask this patient to complete the course of antibiotic. Okay, and also, whenever we shouldn't use the same antibiotic every time. Again, because if you're using the same antibiotic every time the person gets the same disease, this may develop also more bacteria resistant. So do not prescribe or do not use the same antibiotic every time, which is called antibiotic rotation. Of course, this advice may be not for is addressed for a student that maybe is going to become a doctor, is going for a medical school. That's why it's good for now, from now to know, it's not good to use the same antibiotic over again and again, because this will develop more bacterial resistance. I hope that this was a well, actually, let me continue, because the question has not finished. We're still having D for the question, so let's also do D. Okay, it's the same question. Now, hormones are used to treat variety of conditions, and then the most common hormonal treatment is the use of insulin to treat diabetes. Basically, this is again touching base on the genetic engineering. And that's very nice question to wrap up today's tutorial, because basically these are the steps of genetic engineering. You need to put them in order, which kind of help the question that I will help to consolidate the questions that I already explained in the beginning of the class. And he helped you with the first step, which is gene for the human insulin found to be on chromosome number 11. Okay, I found that gene, thanks God. Now, what am I gonna do? What am I gonna do next? After I found this, I need to remove it. I found the gene, thanks God. Now let's remove this gene from the human cell, from chromosome number 11, which is P, right? Then after I remove it, I need to have this gene inserted. So after it's removed, it's going to be inserted into the plasmid vector in the bacteria, right? So that's V. And then I need this plasmid vector to enter the bacteria. I need to insert it out again inside the bacteria, which is R. And then this bacteria will divide by binary fission. Binary fission is a kind of sexual reproduction. As we mentioned, that's a very fast way for reproduction of bacteria. And then this bacteria is going to use this insulin recipe to make more and more insulin. And let's move to the last part of this question. Diabetes is often treated with human insulin that has been produced by genetically modified cells. In most countries, this type of insulin has replaced the insulin that was prepared from animals. Suggest the advantage of providing human insulin to people with diabetes rather than insulin obtained from animals. This is also touching base about immunity. Why is it better to use insulin from like human insulin rather than insulin obtained from animals? Now, first of all, it's easier. Okay? It's easier. Not only easier, it's also quicker. Why? Because the method that we explained, the genetic engineering, the first advantage we said that it's quicker. Remember, we said sexual reproduction is very quick, very fast. So this is very, very fast. Now, the second thing that rather than this genetic engineering method is quicker and faster, also basically or most probably the human insulin would be more effective than for us, right? More effective for us than the animal insulin, because we're human, right? And we're getting insulin from the animal insulin. So it should be more effective for us than the animal insulin. Now, also, if I'm gonna touch base on immunity, there should be less rejection or no rejection, right? Because we're having more common allergens, or we're having more common cell structure or antigens with human cells rather than with animal cells. So it was have less rejection. It would cause less allergy, definitely. Okay, because we're kind of more similar, have more similar structure than the animals. And using also animal or insulin from animals, sometimes it raises ethical point. Ethical point is always accompanied with using animals for medicine, for clinical trials. Okay, so always think of the animal welfare consideration, and try to, there will be always an ethical part and animal welfare consideration that we should refer to. Okay, someone will ask, but when I'm using the human insulin, isn't that ethical part for human? No, because you're just removing the gene out of one of the cells, and then you're using bacteria to reproduce the insulin, just the way I explained here. You're basically not using human. You're just taking part of the gene and then inserting it inside the bacterial vector to get the human insulin. This is how genetic engineering is working. You're not getting human insulin and then injecting it directly into people. You're using genetic engineering to make it. Okay. I hope this class was useful for everyone. This is one of the hard and challenging topics, and I will keep on posting some challenging parts videos in the coming days. Stay tuned. Thank you. Bye.